Yes, so the northern blot hybridization is just as same as the southern blot hybridization. The process-wise, it's the same probe, but only the target molecule is different. So pre-hybridization, hybridization, X-ray film detection, everything is identical. So I'm going to show you diagram. Yes, the electrophoresis, blotting, pre-hybridization and hybridization is all the same. The only difference is what target molecule is being separated on a gel through the electrophoresis. It's RNA in this case. Of course, you need to be careful with dealing with RNA because they are degraded very often, easily. So RNA's inhibitors should be treated with the pipette tip, all these electrophoresis equipment, buffer, water, and everything should be DPC treated, all baked at 300 degrees for 6 hours, right? That may be the difference. Okay, and then detection is same, X-ray film, where that RNA binding with your probe is located. Okay, so then you may wonder, then what are we doing this northern blot hybridization for? Think about it. So presence of a certain mRNA molecule within the cell, what does it tell you? Okay, mRNA molecules are being made from your DNA, the gene, right? Gene-specific transcription will take place. You will make that mRNA, and that mRNA will be used for building proteins. That means that gene expression process is happening. It's being initiated in the cell. It's happening within the cell. So what does that tell you? Yes, you can tell if certain gene within your genome is expressed or not by doing this northern blot hybridization process. So what does that mean? You may have 30,000 genes within your genome. Okay, are they turned on all the time? The answer is no. The genes are highly regulated. There are certain conditions or certain signals that only selectively turn on certain genes. Then you may wonder which genes are turned on under what circumstance, under what signals. That is what this process is answering. Northern blot hybridization will answer you under what condition your gene will be turned on. Think about it. When your cells are exposed to a certain condition, you may wonder, is this condition enough to turn on my gene that I'm studying? Yes, you can answer the question by doing this northern blot hybridization. Okay, does that make sense? So DNA microarray technique. So in the previous lecture 11, I show you the process that allow you to build gene chip, right? So gene chip will have bunch of oligonucleotide that the probes embedded on a little chip. Okay, you're going to use that chip to assay if certain genes are being expressed or not. So it's called microarray technique. Microarray. Okay, so let's look at this. So you can build those gene chip. Okay, little slide, a bunch of nucleotide, all the probes embedded on them. Okay, on those probes, then you can add so-called RNA-based molecules, cDNA molecules, right? Instead of just adding RNA molecules and doing the hybridization here, instead you want to convert that RNA molecules into a more stable form of nucleic acid. Those are DNA molecules. Okay, so you can build DNA out of RNA. We call it reverse transcription. Okay, so when you do the reverse transcription, you are going to use nucleotides that are normally labeled with fluorescence. Okay, so we're talking about here. So RNA molecules are isolated from the cell. Okay, so you have RNA molecules. It's the starting of the northern blot hybridization, right? But instead of using these RNA molecules for your northern blot directly, you add one more step. That is making DNA copies of your RNA. So we call those DNA copies as cDNA molecules. You can do that using a special enzyme called what? Reverse transcriptase. We talked about reverse transcriptase when we talked about RT-PCR. Reverse transcriptase PCR. Yes, you can do that. So when you do that, can you build cDNA molecules? When you do that, add nucleotide that's labeled with certain fluoropole, maybe red fluoropole or green fluoropole. So if you do that, then yes, your cDNA molecules will be labeled with certain fluorescent dye, maybe red or green. Now you have these cDNA molecules, right? Now those cDNA molecules will be hybridizing to the gene chip that has hundreds of those probes embedded on there. So let's say, one probe is binding with this cDNA, which was built based on, these RNA molecules that are isolated from this B cell. Let's consider that B cells are normal cells, but A cells are cancer cells. Okay, so we are asking, okay, is there a difference in the gene expression pattern between the normal cells and cancer cells? You can answer that by doing this process. So all the RNA that are being made from normal cells and cancer cells are isolated, right? And then those RNA molecules will be all reverse transcriptized into cDNA molecules. cDNA molecules, cDNA molecules. So green cDNA molecules are the cDNA molecules made from normal cells, okay, because they were made from the RNA molecules that are made from normal cells. But red cDNA molecules are made from RNA molecules that are isolated from cancer cells. Okay, you see the differences, right? So those cDNA molecules now are hybridizing to the probes that are embedded on your gene chip. Okay, if there's a difference in the binding, okay, so let's say these cDNA bind on that probe, but these didn't bind, okay, then you will see green spots there. If it's the opposite case, if these cDNA bind on that probe but none of these bind, those will be red. Okay, if both of them bind, green and red together, maybe it's some kind of mutual color, maybe yellow. Okay, if none of these cDNA bind on the probe, there's nothing. Okay, so you will get about 3 different signals. Okay, maybe green, red, or yellow. Okay, so by looking at that, what can you tell? Whatever that gene probe is, that gene is not being expressed from neither cells, right? And if you see yellow, what does that mean? Whatever that gene probe is made out of, that gene is being expressed in both cells, normal cells and cancer cells, right? If you see red, what does that mean? That gene is only expressed from the cancer cells. If you see green, that means that gene is only expressed from normal cells. Okay, so that is the kind of result of difference in the gene expression pattern. Does it make sense? So you can look at that. So you can also apply this animation that shows you the process microarray by copying and pasting that address on a new browser. So let's say if you can paste that and enter, then yes, microarray methodology flash animation. So click here, allow that, yes. Okay, just go through this animation, it'll tell you what's happening. So let's go, and common DNA chip is determining which genes are activated and they are being expressed. Okay, so let's talk about E cells, okay, that are being exposed to oxygen present condition, that are being exposed to non-oxygen condition, anoxic condition, and you're going to compare their gene expression pattern. Okay, so let's do. So E cells, okay, then one is open, so oxygen is present, the other is closed, so they don't have any oxygen. Okay, is the difference in their gene expression pattern. It's loud. So after a certain growth period, now you're going to isolate their RNA molecules to see their different gene expression pattern. Cells are there. After centrifugation, you're removing the medium. Okay, and then what? You're going to add certain enzymes to break open the cells. And then you are extracting RNA molecules from each cell type. Okay, then what? Let's see. Okay, RNA molecules, right? Long poly A tail. Do the reverse transcription. Okay, that's the reverse transcription. With the long T tail. Now these are cDNA molecules that are being made. When you do that, you add a nucleotide with green fluorescence, nucleotide with red fluorescence. Now they are differently labeled, right? And get those transferred to a maybe tube. I'm going to mix them up because they are differently labeled. That's fine. And Then you're going to add them to the gene chip that has a bunch of the probes embedded on there. Okay. cDNA is being added to the probes. Now see if they will bind to specific probes. Okay, if they do, that means what? cDNA is binding with certain probe shows the hybridization. If that's what you see, what does that mean? Okay. Green fluorescence on both, red fluorescence on these. Okay. So whatever that gene is, it's being expressed under anoxic condition and normal condition. That is only expressed on the normal condition. That is only expressed under the anoxic condition. That's what it means, right? There you go. Okay. So now you can understand the difference in the gene expression pattern using this microarray and gene chip technology. So that is basically the same as nodal blot technology, but it's much more efficient because you're handling about hundreds of probes at a time. So you're hybridizing hundreds of probes in a short period of time. That's more advanced form of nodal blot analysis. Okay, so that's it.